This week in lab, you will be using your lab kit to study enzymes. And you're going to be looking at some different factors that affect enzymes, including the concentration of the enzyme and the temperature of the enzyme. So let's start by looking at the materials that you need and where you will find them in your kit. So first of all, I want to point out that I have my countertop covered. I actually have a plastic bag, just a trash bag, covered with an old towel. We will be working with hydrogen peroxide, which can bleach out and potentially damage surfaces. So you might want to just get an old towel or some sort of plastic to put down just to protect your countertop or whatever surface you are on. In addition to that, you need all of the materials that are in the bag that's labeled Enzyme Catalysis. And in this bag, you will find 2 medicine cups, and I've already labeled them. It'll tell you in the procedure to label one as 1.5% hydrogen peroxide and the other one as 100% catalase. You will also need the thermometer, yeast, which is going to be your source of catalase, a petri dish, which you can see that I already have some little circles in there, and I'll explain how I got those in a moment. 2 transfer pipettes, one of them labeled H2O or water, the other one labeled catalase. And then filter paper. You should have 3 pieces of filter paper. And you can see that I've already used one of my pieces to cut out some circles. In addition to that kit, you also need to get the Principles of Biology equipment set. And you will need some materials out of this bag as well. You will need a 250 milliliter beaker, some forceps, also known as tweezers, the 25 milliliter graduated cylinder. So this is the smaller. There is a larger one in there. You want the smaller one. A ruler, 5 test tubes and the test tube rack, and a grease pencil. Additionally, you will need some materials from the chemistry safety pack. As you can see, I'm wearing this nifty apron, which is going to protect my clothing. You will also need the goggles and a pair of gloves. In addition to the materials that are in your kit, you also need to supply some materials that either you will have hopefully around the household or you may have to go buy them. So first of all, you're going to need some purified or distilled water. You can use bottled water such as Dasani or Aquafina. I have a gallon here, but you don't really need a whole gallon for this lab. Probably just a regular sized, 16 to 20 ounce bottle of water is sufficient for today's lab. You also need some hydrogen peroxide. This you can buy at any grocery store or like a Walgreens or any drugstore. It's probably about $1 for this, so it's not too expensive. If you already have some hydrogen peroxide in your house, it's probably the correct stuff. If you look on the back, it should say 3% hydrogen peroxide. You only need 20 milliliters, and this is about 1,000 milliliters. So even if you have an open one at home and you only have a little bit left, it's probably enough for this week's lab. You will likely also need a pair of scissors. You will need a teaspoon. And because I don't really like the grease pencil that they give you in the kit, I also grabbed a Sharpie, so you might find that helpful. You should grab your phone. You will need your phone for a couple of reasons. You will need to time some things, and so you'll use the timer on your phone. You can also use a stopwatch or really anything that has a timer, but your phone might be the easiest. It just needs to have a stopwatch function that will count up. Oh, you are also going to be writing a lab report on this week's lab. So I recommend that you take pictures as you go through the activity to help you remember what you did for when you write the lab report. And then you also need a hole puncher. If you have a single hole punch, that actually works better. I don't have a single hole punch, so I just use my 3-hole punch. And that is used on the filter paper. And that's how I got these little circles here. And so I just popped the back off and then used one of them to cut out the circles that I needed. If you don't have a hole puncher, you can just cut your filter paper into squares. It doesn't have to be round. Squares are fine. You just want to try to make sure that each square is the exact same size or as close as possible. So you'll want to measure it out. I believe in the protocol it says about 6 to 7 millimeters, which are going to be the smaller ones on the metric side of your ruler. So for prep work, like I said, I use a Sharpie, or you can use the grease pencil that they provide. As I mentioned on the medicine cups, you want to label one as 1.5% hydrogen peroxide. One of them should be labeled as 100% catalase. And the test tubes should be labeled as 0%, 25%, 50%, 75%, and 100%. Additionally, you will need 20 circles punched out of your filter paper, or if you're cutting it into squares, you will need 20 squares. Okay, so now that we have all of our materials, let's go ahead and mix up our catalase solution. Now, catalase is going to be the enzyme that we're using for the lab, and we are getting our catalase from yeast. So we can also get catalase from different sources, but yeast is a pretty simple and easy way to get it. So to make up your catalase, you want to fill your beaker with about 250 milliliters of water. You can use tap water for this. And you want it to be around 35 degrees Celsius. So in your materials, you should have had a thermometer. And the thermometer only measures in Celsius. So you don't have to worry about whether you're looking at Celsius or Fahrenheit. And so you just turn the warm water on, and you can just run this right under the tap. So I've already done that, but just to double check. So yep, I'm right around 35, so I'm still good. So once you have your 250 milliliters of 35 degrees Celsius water, you will use your teaspoon and your yeast packet to measure out 1 teaspoon of yeast and add it to your beaker of water. You can use the tablespoon or some other device to stir the mixture. Maybe remove your thermometer before you add the yeast, or else it's going to make a mess. But go ahead and give that a nice stir to mix it up to make your catalase. All right, after you've done that, you are going to get your medicine cup that is labeled 100% catalase, and you're going to fill it up to the 20 milliliter mark with your catalase solution. In the other medicine cup, labeled 1.5% hydrogen peroxide, we are going to add 10 milliliters of our 3% hydrogen peroxide and 10 milliliters of our purified water. Now, I poured my purified water into a measuring cup just to make it easier to work with, since mine was in a whole gallon. But remember, you can also just use a water bottle as long as it's bottled and purified. Now, when you're measuring something in a container like a graduated cylinder, you have to be mindful of the meniscus. And what the meniscus is that the liquid tends to kind of stick or crawl up the edges of the container, and you end up with this kind of dip in the liquid. And so to accurately measure liquid, you need to look at the bottom of that dip. So if it's like a U shape, you want to measure from the bottom of the U. So we're going to start with our hydrogen peroxide and measure out, oops, this is not going well. Measure out your 10 milliliters. And it's best to measure the meniscus by putting it flat on the surface and getting down at eye level. Once you have an accurate measure, make sure you go ahead and close your hydrogen peroxide to prevent spilling it. So you are going to take your hydrogen peroxide and pour it into that medicine cup. And then you're going to use your room temperature purified water. And once again, fill it up to that 10 milliliter mark. And once you have an accurate measurement, you can go ahead and pour it into the medicine cup. And now you have your diluted hydrogen peroxide solution. So now you are going to use your test tube holder, and you should have 5 test tubes in it, labeled 0, 25, 50, 75, and 100. And what you're going to do is take your catalase, and we're going to be diluting it to these different percentages. So we're going to be testing the effect of different enzyme concentrations on the reaction rate. So to get those different concentrations, there is a chart that tells you how many drops of catalase and how many drops of water to add to each of these test tubes. Now, when you're adding the water, you want to use your room temperature purified water. So using the pipette labeled catalase, you are going to add the catalase solution and add the proper number of drops based on the table into each of the test tubes. Now, it's important that you kind of stir the catalase solution before you start aliquoting it out into your test tubes, because it tends to kind of settle and you get a separation of the materials. So just make sure, and you can just use the pipetter just to stir it really well. And in the 0% tube, you're not going to add any catalase because that's just going to be pure water. In the 25% tube, you are going to add 5 drops of catalase. 1, 2, 3, 4, 5. In the 50%, you're going to add 10 drops of catalase. In the 75%, you're going to add 15 drops of catalase. And in the 100% catalase, you're going to add 20 drops of the catalase. Now to add the water, you're going to use the other transfer pipette that's labeled as water. And once again, remember you're using your purified water. So to the one labeled 0, you're going to add 20 drops of water. To the one labeled 25, you're going to add 15 drops of water. To the one labeled 50%, you're going to add 10 drops of water. To the one labeled 75%, we're going to add 5 drops of water. And to the one labeled 100%, we are not going to add any water. And for each of your test tubes, you want to make sure that you swirl it in order to mix the contents really well. So now you can set the catalase aside and you can set the water aside, and go ahead and grab your 1.5% hydrogen peroxide solution, which is in that medicine cup. And what you're going to do is you are going to use the forceps, otherwise known as tweezers, and you are going to grab one of the circles or the squares that you cut out of the filter paper. These, you have to make sure that these little parts line up or else they don't grip very well. So just be mindful of that if you run into problems. Now you want to have your timer ready to go. So I will pull up my phone and use my timer. Remember, you want to do the stopwatch function because you want to count up from 0, and you're going to time how long it takes this little disc of paper to rise from the bottom of the medicine cup all the way up to the top. But before you place it in there, we're going to dip it into the different catalase concentrations. So always start with the lowest concentration first. So we're going to start with the 0% catalase first, and then move up to 25, and then 50, and then 75, and 100. And we're going to do at least 2 trials for each concentration. If your 2 trials differ by a large amount, I think it says more than 4 seconds, then you should do a 3rd trial and then pick the 2 closest of the 3 trials. So to do this, you'll probably need to dip your test tube off to the side so that you can get the little disc wet. And this is a little tricky. So you want to get it wet, but you don't want to pour it out. Go ahead and drag the filter paper on the edge of the test tube just to get the excess liquid off. And then start your timer. As you place it on the bottom. And then just wait for it to rise to the top. And it says that if it doesn't rise to the top within 3 minutes, then you can just stop the timer. I'm going to try this one with the 25% catalase, but I can give it a nice little mix before I dip the disc in it. Remember to wipe the sides off on the test tube just to get the excess off. Get your timer ready and place it at the bottom and then start the timer and see how long it takes for it to rise to the top. So in between each trial, make sure you remove the old disc before putting the new disc in. And at the end, it tells you to measure the height of the hydrogen peroxide. So you can just use your ruler, and you want to make sure you're using the metric side, so the side that has centimeters, and you want to measure the height of the liquid in your medicine cup. So that is how you do the different concentrations of the enzyme, which is the catalase, which is that yeast and water mixture. Now, for the second part of lab, you are going to be testing the effects of different temperatures on this reaction as well. If you're doing it right away, you can use the same catalase and hydrogen peroxide solutions that you've already mixed up. If you are going to do it later, then you'll need to remake these, but you should have plenty of supplies in order to do that. So I will let you read through the protocol for the temperature part of the lab. It's going to be very similar, but it's going to tell you to use 100% catalase solution. You're going to reuse these same test tubes. So you're just going to want to rinse them out really well in the sink and just follow the instructions on how to essentially alter the temperature. So you're going to need to have some hot water and some ice. If you don't have crushed ice that you can get out of your refrigerator, you can maybe just take ice and put it in a plastic baggie and hit it with like a rolling pin or a hammer or something just to kind of crush it up. And you'll need that in order to make the enzyme colder. And then you'll also look at it in a hotter environment by placing the test tube in some hot water. As a reminder, you will be writing a lab report on this lab, so make sure you take careful notes as to what you did. You might also want to take some pictures so that you remember the setup for when you write your lab report. In addition to recording your data, you should record anything that maybe didn't work out as expected. So if something goes wrong or you weren't able to perform one of them because of, some reason, let's say one of your test tubes, you knocked it over and spilled it and you couldn't make up the concentration again, that's a good thing to note as well. And you can include that in your lab report as well. So good luck with your experiment. Let me know if you have any questions as you are performing it, and I would be happy to help.